So we have, Randy, about 5 minutes before we're going to start. I don't know. I thought folks might be interested in the fact that you're in California, which is now based on a number of issues, primarily climate change,
Yes.
is experiencing some of the worst wildfires in California history. So we here in New Mexico, we're no strangers to wildfires. We had a big 450,000 acre wildfire a couple of years ago. And as I mentioned, I was evacuated from Los Alamos 2 separate times, big wildfires, one that came into town and burned out 350 homes.
Oh, man.
And the second one that they kind of were able to keep it at bay. So I'm sure folks would be interested and concerned. How are things going in California from your perspective?
Well, I haven't seen the update this morning for the Dixie Fire, but the night before last, it expanded by 110 square miles overnight. 110 square miles from the evening until morning. It was mostly national forest land. We have a lot of national forest land in California. Most all of our forest land has been mismanaged, not intentionally, with fire suppression over the years. During the gold rush, the forests were cut down extensively, and we now have second or third growth forests, which and then we also the Indians who managed our forests for many years with controlled burns, that process stopped. So now we have these monster fuel loads, and then with climate change, which we are experiencing tremendously, the different ecosystems are changing, moving uphill. The traditional pastures of native vegetation that beekeepers have gone to for generations, they don't exist anymore. They're all moving uphill. So we're having huge vegetation changes. And then we have this mega drought, which we're having whole swaths of forest just drying up and dying. So trees that are 30, 40 years old that obviously have not seen this kind of drought are just dying from lack of water. So we live essentially in this tinderbox. And as I was telling a friend recently, this is not going to end until all these forests come back into balance and these huge fuel loads get taken care of. So we had Monday of this week, we had a local fire, a fairly small one, approaching our bee yard. It was also approaching the air tanker attack station, so they prioritized it. And we talked to the pilot afterwards. He saw our honeybees there, our bee yard, and he laid a swath of the Fostech down right halfway on the beehives and halfway on right next to them, and that stopped the fire right there. So we saved that yard of bees intentionally.
Oh wow!
But we have 60 out yards in our county, and so anytime there's any fire, we often have an out yard threatened. So we've been pretty lucky. We've only lost one so far last year, and we'll see what happens.
Well, that's really something. There's kind of that. Well, here in New Mexico, we have the same issues with wanting to go in and do these prescribed burns, to try to clear out some of that underbrush that's been laying there for so many years. And so it's a little frightening for people, because that's how the very first Los Alamos fire started. They did the prescribed burn on a windy day in the spring, and the prescribed burn went out of control. And that's when
always an inherent risk with prescribed burns.
Yeah. And so we have great weather here in New Mexico, but we have pretty heavy winds in the spring. So I just kind of wish they would not wait till the fall to do these prescribed burns. But everyone here is, and maybe it'll happen. Maybe it's the same way in California, but when you see smoke, everyone sort of, there's a website to go to that will let you know pretty much, okay, don't worry, that's a prescribed burn. That's an intentionally set fire, and hopefully it's under control. But it's kind of a balance, because in fact, we have these smoke jumpers here in New Mexico called the hot shots. And I believe the hot shots are actually in California because of the struggles you're having. We're actually having a pretty good monsoon season. sort of unusual because the past few years we have what's called the monsoon, where it never shows up. But we've actually been getting some pretty good rain, so we're lucky. But we realize that at any given year, it can change back to a serious drought.
Right.
So very glad that you're here with us. It's now 9 o'clock. So let me, if I can, do a brief introduction of Randy Oliver. Most people know you, Randy, and we're so glad to have you back. It was one of the highlights of our winter conference were your 2 talks. But Randy Oliver is a beekeeper who's kept bees for most of his life, and he views beekeeping through the eyes of a biologist, a researcher, and a nature lover. He makes his living as a commercial beekeeper and maintains the website scientificbeekeeping.com. He's a popular writer in bee journals. He's almost every month, he's in American Bee Journal, and he's an invited speaker worldwide. We're really glad that you're here, Randy. We're really glad the fires are somewhat not impacting you. He loves sharing his knowledge of bee biology, colony health issues, and a practical beekeeping management. And I know that you're a generational beekeeper in that your sons and even your grandson has gotten involved in beekeeping. And so that kind of warms my heart when I see those pictures of that little guy taking notes in the bee yard. It's just wonderful. So thank you so much for being here. I hope that things continue to go well for you. Anyway, we're going to see you twice today. You sort of bookend our conference, and we're very grateful and glad to see you. So hopefully everything's set up so that you can share your slides.
Okay, let's try it here. Here we go. And let me slideshow from beginning. There we go. All right. Okay, so let's just get going here. There we go. And what I call this is reading the combs. And the point is that you're going to find plenty of people who will tell you how to keep bees. And I'm not going to at all. I don't make recommendations, but I will describe the biology of bees and then allow you to make your own management decisions. And the way you can do that is by reading the combs. Let's start off first. I'm assuming you're going to have some beginning beekeepers in the audience. And if you do a Google search for beekeeping, you're going to get 32 million results. After you've read the first million or so of those, you're going to realize there's lots of very strong opinions in beekeeping, and many of them are very much conflicting. This brings you back to a couple of articles, scientific articles published by Denning and Kruger. They published it in 1999, then again 10 years later in 2009. It's called Unskilled and Unaware of It: How Difficulties in Recognizing One's Own Incompetence Lead to Inflated Self-Assessments. And so this is essentially the Dunning-Kruger curve I've drawn right here. And it applies exactly to beekeeping, whereas the y-axis is your self-assessment of your own knowledge and ability. And the x-axis is how many years you've been keeping bees. And I mean serious beekeeping, not just poking the hive every now and then, but really spending a lot of time in the hives. And you start off with your first hive on foundation, maybe give them a little sugar syrup, get that first honey flow, they build up, they do great. And at this point now, you are at your peak of self-inflated assessment. You're thinking, oh my God, this is so easy. I don't understand all these problems. I must be the smartest beekeeper in the world. And this is where a number of people start their beekeeping blog right here. And then they fail to control Varroa adequately, and they don't allow enough winter stores for the colony, and the colonies all die. At that point, you hit the trough of disillusionment. At this point, traditionally, we've always had a lot of beekeepers drop off at this point. A lot of them just give it up. But some will get back on the horse and restock those hives, and they get on this slope of experience and enlightenment about beekeeping, and finally attain the slow mastery of the craft. There are a number of beekeepers that are first-year beekeepers for 20 years in a row. They just never seem to learn at all, and they have problems year after year. But the rest of us start to learn with experience. And it's very humbling with the bees and to do that. The key thing here is that this slope never plateaus. you have a chance of learning for the rest of your life. I have more questions about beekeeping now than I've ever had in my life. And I spend my life answering questions. I have 4 field trials going right now. Today, I'll be out working on 2 of them. Tuesday, we take a bunch of data, answering questions that I have about beekeeping. So this, to me, is very exciting. This is something you can do for the rest of your life, asking these questions. So rather than asking beekeepers or books what the rules are, I thought I'd give you the rules of beekeeping from the honeybee's point of view. And here's the key thing that I found. If you want to be a successful beekeeper, stop looking at the world through the eyes of a human being and start looking at the world through the eyes of the honeybee, so that the more you understand how the bees see the world, the more you understand how to work with them. So here's the rules. Number 1, they need a warm, dry cavity. They evolved in Africa, in tropical areas, and just as humans, who also evolved there, moved into more northerly climes by living in cavities, in caves, or in man-made structures. The honeybees have been able to adapt to cooler climates by living in mainly hollow tree cavities and storing food for the winter. From a bee husbandry standpoint, this is pretty simple. Give them a dry box. The bees do not care if they're in a Langstroth hive, or a Wari hive, or a top bar hive, or an 8-frame hive, or a 10-frame hive, or whatever color is painted. They totally do not recognize the type of hive at all. All they care about is this warm, dry cavity. So this is an easy thing for bee husbandry to provide. Number 2, they need food. Natural food of honeybees are nectar and pollen. Nectar being the carbohydrate source, and pollen being the source of the protein and sterols and lipids and vitamins and minerals. So if you're in an area where there are deficiencies of nectar and pollen, you may not be able to keep bees there. Or what many migratory beekeepers do is, when things dry up during certain periods of the year, they'll put their hives on trucks or on donkeys or whatever, and they'll move them to areas where there's better forage. The other alternative for bee husbandry is, if necessary, to provide them with supplemental nutrition. So if they don't have enough stores for the winter, you can substitute that with sugar syrup. If you feel that they are short on protein, you can supplement it with pollen subs, which are improving year by year. So again, for the actual husbandry, you can always step in, either move the hives to better forage or to supplement their nutrition. And rule number 3 is that every species has parasites. Every human being has parasitic mites. You all have parasitic mites in your hair follicles. But you have a good host-parasite relationship, and most of you never in your life will notice that you have parasitic mites living on your body, in your hair follicles. The honeybees have a new major parasite. They picked up a few of them in recent years, but this is the Varroa Mite. And the Varroa Mite transmits a virus called deformed wing virus. We're witnessing this evolutionary transition in the honeybee population now, from being naive to this virus and this vector, the Varroa Mite, to eventually we'll have all resistant stock of bees. But you are lucky to be able to watch this biological transformation right now. Unfortunately, many thousands of colonies are dying during this transformation, as nature and the bee breeders start to weed out the susceptible stock of bees and select for the resistant stocks. From a management standpoint, unless you are keeping mite-resistant bees, bees that have genetically programmed to show abilities to resist the mite or to tolerate the virus, you need to intervene to help your bees deal with this mite. Otherwise, your colonies will typically die. And that's the end of the rules. So we can go back over them very, very quickly. They need dry cavity, they need food, they need parasite management. After that, pretty much anything anybody tells you as far as what you need to do as a beekeeper that's a reflection of that person rather than what the bees need. Here's my simple recipe for healthy hives. I'll repeat this at the end here. First thing, keep young, vigorous queens. In nature, in areas that have a relatively short winter, queens typically live about 18 months, and then they get naturally superseded. We all know that with human beings, the females improve with age. That is not the case with honeybees. The younger queens are much more vigorous, and the colonies generally do better with younger queens. So requeening each year helps a lot. Provide adequate nutrition. I talked about that. Control Varroa. Don't wait until Varroa levels get high. Control them all year long. Keep the mite count low all year, and your bees can thrive. Minimize toxins in the hive. That's not only man-made toxins, but natural toxins. We have 2 very toxic plants here in California that can wipe out hives. Then there's the agricultural toxins, the pesticides. And then there's the ones that the beekeepers, well-meaning beekeepers, dumping things in the hive, essential oils, all these home remedies. Most of those are very stressful for the bees. So be careful what you put in your hive, especially things that leave residues in the combs. And the most important thing is to be proactive rather than reactive. The proactive beekeeper foresees problems coming down the pipe and manages his hives to avoid those ever becoming a problem. Whereas the reactive beekeeper is always dealing with the problem reactively, and that's much more difficult to bring the hive back to health than it is to keep it in health from the beginning. So my suggestions, number 1, honeybees are fascinating, eusocial animal, but they're still just animals, and all the principles for animal husbandry for any other type of animal apply to the honeybee also. Second, copy consistently successful local beekeepers. I'll tell you right now, I recently I'm recovering from chemo radiation therapy for a tumor on my tongue, which wiped out my salivary gland. So I need to get a sip of moisture frequently, or my lips glue to my teeth. Okay, consistently successful is the key word right here. And the way you can tell it's consistently successful beekeeper, it's not that they are the a leader of a club. They're not the ones that are necessarily interviewed by the media. You can tell them because their main problem every spring is they have too many bees. They need to get rid of bees. They got to give them away. They got to shake bees and sell them. They got to sell nukes or something like that. Any beekeeper who is buying bees in the spring to replace losses is not a consistently successful beekeeper. So look for those as a mentor. Look for a beekeeper whose main problem is having too many bees every spring. Number 3, know the 4 horsemen of bee apocalypse, and those are any kind of chilling. So bees again are a tropical animal. So whenever the conditions are cold, that is stressful for the bees, makes them susceptible to some of their parasites. Poor nutrition is huge. The immunocompetence of bees and the ability for colonies to grow is all dependent upon adequate nutrition, especially the amount of high quality pollen coming in. Toxins, already discussed that, both natural and made toxins. And then parasites. There's several of them that can cause disease, but the main one we're dealing with these days is the Varroa Mite. It used to be just the American foul brood bacteria. That's all we ever talked about for bee disease for many, many years, but now it's mostly Varroa. Then suggestion number 4, on all your management decisions, rather than trying to follow a on the back of the box recipe for making brownies or something, don't look for a recipe. Look at the colony, at the biological needs of that colony by opening it up and actually looking at the combs, and then base your management decisions upon what you see by looking at those combs. And that's where I get this this term, learning to read the combs. So that's what the rest of this presentation will be about, how to read the combs of the hive. And what I like here is this is a quote from Ticknor Edwards back from 1920. And what he said is, and so the best bee master is always the one who most exactly obeys the orders from the hive. And as we were setting up our fair booth yesterday, our exhibit there, I was explaining to one of my helpers that you're not going to ever change one tiny thing about bee behavior. That's fixed genetically. So what you can do is the more you can understand bee biology and bee behavior, the more you can make it look like magic, and the more you can make it look like you're controlling anything, but you don't control anything with the bees. The bees their behavior is fixed. So the more you can be a servant to the bees and understand what the bees actually need, the better beekeeper you can be. So let's look at the business model or the ecological niche of the honeybee superorganism. It's a highly efficient competitor for sporadic resources. When flowers bloom, these guys are competing against all the other pollinators, and they have developed strategies to outcompete many other pollinators. There's only two main objectives for the bee colony. One is to store enough food in the form of honey. And this is why it's called the honeybee. Don't ever forget that the number one thing for honeybees is to store honey when it's available for reserves for when there are dearths afterwards. The second thing is, once they have adequately filled the cavity and gotten enough food for their own survival, there's only one other thing to live for, and that is to reproduce. And they do that by sending out swarms, as many as they can a year. So once they fill the cavity, they're going to send out swarms. If you understand that these are the two main driving principles for honeybees, then these are what you really want to focus on. The way bees are able to get the jump on their competitors is they have this standing army of foragers just waiting all the time. And all these forager bees, they don't waste energy. They don't fly out looking for anything until the scouts tell them that there is something out there. So they don't waste, the colony does not waste energy. And the foragers are considered to be very expendable. They live very short lives, only 8 to 12 days typically, once they begin foraging. So they just sit on the combs waiting every morning for the scouts to come back and say, yeah, the food is out here, here's where it's at. And then they work hard at bringing that back. The members of the colony inside the box where it's dark, they communicate mostly by smell and by passing food. They have a communal stomach. And they all share food or starve equally. And here's a colony in the process of starvation right here. They've used up their honey reserves. They've used up any pollen reserves, and they've now cannibalized some of their brood. They'll leave the capped sealed brood that does not need feeding to survive, to take their place should they starve. But they all will starve equally other than the queen. They'll try to maintain the queen to the very end. Now, the queen, of course, is very important in the colony, and most people focus on the queen as being a special bee. And that's totally the reverse of the fact. The queen is a normal female hymenopteran insect. She can reproduce, she can lay eggs. It's the workers that are these special bees, these voluntarily sterile females that will never reproduce. These are the special bees in a eusocial insect colony. The queen is just a normal reproductive female. But in the colony, she does serve the function of being the ovary of the colony. She's the only egg layer in the colony. Plus, she's the cohesive core of the colony by her distribution of pheromones that keeps the colony homeostasis working. Other than that pheromonal influence and being the ovary, she has very little influence in the colony whatsoever. The bees that actually run the show are the nurse bees. These are the bees from about 3 days of age to about 15 days of age typically, and that varies quite a bit. But they're the ones that control the food supply, or the protein supply within the colony. They're the only bees that will that consume the incoming pollen or the stored pollen and then convert it to jelly. Jelly being critical in the colony. And what I did is I did a graph here of colony population right here on the y-axis. So this is the colony population building up and then declining during the dearth period. And notice I didn't say winter, I said dearth period. This is the nectar and pollen availability each season. And you can see that the colony population lags about 3 weeks, about one brood cycle behind the curve for the nectar and pollen. There's no counter dates because if you live in the northern hemisphere, you tend to think of winter, of short days being associated with this dip in the colony population. But if you lived over in Southern California or the California coast or Mexico, Then springtime starts in November when it starts to rain, and then the plants can start to grow and the flowers can bloom. Springtime buildup occurs over the winter. Main nectar flow. You have a big one in November, and then you'll have another one perhaps in February, March. And then the dearth period begins in early July and extends until the next November. So during some of the longest days of the year is the dearth period, and the warmest days of the year. So the dearth period is not about day length, and not even so much about temperature, as long as it's warm enough for the bees to fly. It's about the availability of nectar and pollen. So let's understand one more term is attrition and recruitment. So attrition is this loss of workers. Workers don't live very long, about 35 days average during the spring and summer. There's very rapid turnover of their population. Recruitment is the way that the colony adjusts its size and its response to any kind of issues out there. That's by, if it can, just recruit a whole bunch of new bees, and it's like it's a constant war going on against nature with the honeybees. And like any army, as long as you can keep recruiting new soldiers, you can keep being engaged in that war. As soon as recruitment falls off, then that's a problem. And you start to lose the battle. So this recruitment is always going against attrition. And when I plot this out here, put numbers to this, again, here's the colony population on the y-axis over the course of a season. And this is your net daily gain or loss. So you can see here, the colony is gaining about 500 to 600 new workers every single day. That's how fast this colony is growing. And then an equilibrium is reached up here, where the attrition comes in equilibrium with recruitment. And then when the queen starts to reduce egg laying, and the nurses reduce the number of brood they rear later in the season, then attrition exceeds recruitment, you're in negative numbers, and the colony population drops. And the purpose for this is to eliminate the extra mouths to feed, because this is when they're storing the honey, and this is when they have to live on that honey right there. Here's an important point. For those beekeepers who are loath to sacrifice 300 bees to do a mite wash to monitor Varroa, the queen is laying about 1,500, maybe 2,000 eggs a day right here. If the net gain is 500 bees a day, that means you have 1,000 bees a day dying of natural attrition. 1,000 bees a day, every day, are dying of natural attrition here. So sacrificing 300 of them in order to keep the colony healthy is like giving a blood sample to keep yourself healthy. Then I'll also use this colorful graph I created from data from Lloyd Harris, who did this massive study in Manitoba back in the mid-70s of marking bees every 12 days, which is the post-capping period, and then looking at the colony population structure. So the total colony population is reflected again with on the y-axis at the top of the curve right here. The amount of brood in the hive is the dotted line right here. Notice that sometimes the amount of brood exceeds the amount of adult bees in the hive. And then the red are the workers that are 0 to 12 days old, orange 13 to 24 days, 25 to 36 days. Very few bees surviving past 48 days during the summer, and then longer lived bees during the winter. And we'll return to this graph over and over again. Most all these graphs in this presentation are either on my website, many of them in the first year beekeeping webpage. What I've done is on my website, I published a series of articles, 13 articles, taking you through all the different biological phases through the season over the course of a year. So you can read any of these in much more detail. We're going to have a very quick version of going through all of these now. One more piece of advice here is, prior to Varroa, I would suggest that if you don't know what you're doing in the hive, just leave the bees alone. They're better off without you. Nowadays, that doesn't hold true. They're generally better off with you as far as Varroa management. Other than that, my suggestion to you is that before you ever take the lid off a beehive to peek inside, to know exactly what you're going to be looking for and what you plan to do if you see it. Have your plan of action in your head in advance. So for a professional beekeeper, a colony inspection may only take 10 to 15 seconds. It's very, very quick to determine what the needs of the colony or whether it has a problem. But don't just haphazardly or randomly start opening and wondering, looking around. Have a plan. Now, for the new beginners, I suggest starting with a very weak colony, a weak nuke, and then get into that every day or two. That's the way you learn. And you're going to make every kind of mistake. You're going to kill queens, do that kind of stuff. But that's part of the learning curve. But your goal is, once you understand seeing what's happening in the hive, is to have a plan before you ever open a hive. Another thing that can help is for your local club to produce a graph like this for your area. And you can put the dates in, but mainly what you're doing is a phenological curve. So phenological means based upon the bloom of the plants here. So if somebody lives at a higher or lower elevation, the dates may be different. And we have yards where the dates within 20 minutes drive home are full months different for the same plant coming into bloom due to elevation change. That would be much the same up there in New Mexico. Here's the frames of bees in the colony. There's your colony population. The swarming happens here. Takes 60 plus days from when the brood nest gets initiated until you reach a swarming population, then typically another 30 to 60 days before the main honey flow. Again, this is very much this one's very much dependent on the location here. So you can put in toxic plant, Buckeye right here, when you have your dearth, when the main honey flow is, when swarming. This is very useful to have a plan then. So then you can overlay what you want to be doing at each time. And again, these graphs are on my first year beekeeping. And what you want to do is for all your management to be proactive, you count backwards from each one of these key points, your main honey flow, swarming. So that means to be proactive, you're looking a few weeks before any of these events to avoid those problems, especially with Varroa right here. Putting in whether you're going to be doing any kind of supplemental feeding or avoiding starvation. So create a graph like this for your local club. It will help everybody a whole lot. Okay, now we're going to talk about actually reading the combs and identifying things. Honey, you can tell whether it's freshly stored honey or honey that's been there for a while by whether there's still an airspace under the cappings. So when it's freshly capped over, there's an airspace beneath the cappings, and the cappings look light colored. When the airspace air dissipates out and the capping comes down, touches the honey, then the cappings look dark. So that means that's honey that's been there for a while. It doesn't change the color of the honey. It's just that the airspace is lost. Then you have fresh incoming pollen, which is dull colored, freshly brought in by the pollen foragers. And then any surplus uneaten pollen for that day gets nectar added to it and packed down and looks more shiny. And that's stored fermented bee bread right here. There's a common misconception that this fermentation improves the nutritional quality of the pollen. I have not seen any evidence in support of that theory whatsoever. Then you have the nurse bees. Those are the ones that are eating this pollen and then feeding it to the brood. You have open brood, which are the eggs and the larvae, and the sealed brood, with the other name for sealed brood being Varroa food. So anytime you have sealed brood in a colony, Varroa can reproduce. So you may wonder, if nurse bees run the show, how do they know what's happening outside the hive? So the view inside the hive is this. It's pitch black. So the inputs to the nurse bees have to be sensual cues rather than such as olfaction and tactile rather than visual. So what they look at is they look at the amount of incoming nectar from the foragers, the incoming pollen from the foragers, perhaps monitoring their honey reserves, that's questionable. And then whether there's larval pheromones from the young larvae, which gives them an idea of what that the queen is laying and that there are brood to be fed. This also is a cue, these larval pheromones that tell the whole rest of the colony whether or not the queen is laying eggs, the E-beta ocimene, young larval pheromone, which is a big cue for swarming or for supersedure. And the place to pay attention to is this interface between the honey. So here's some fresh honey, still has the air spaces right here. Whether or not you have a band of unconsumed pollen right here. This is the pollen reserve right here, and then the brood. So this interface gives you the best idea of what's happening in the hive. So I consider these to be the energy stores, this honey right here. The bee bread is any protein reserve, leftover protein that wasn't consumed that day. And then your brood is your energy and protein demand, the main demand down here. The energy interface is this band of cells right along here, which will either be filled with honey, freshly uncapped, if the bees are going into their stores, eating their way up, or it'll be nectar ripening to be stored, meaning that the colony is expanding downward, backfilling, and going to be pushing the brood nest downward. So the energy interface. Then you have your protein interface. So if they're living hand to mouth, eating all their pollen, you won't see a surplus of bee bread here. If they have excess, then you'll see this band of pollen right here. So this protein interface tells you a lot. And here's just to show that the nurse bees are the only ones that eat pollen. There's 2 squashes in Ziploc bags where I'm squashing for nosema analysis. If you squash nurse bees and mix them with water, you'll see all the pollen from their guts turn the water colorful, the color of the pollen. With foragers, you do the same thing. There's no pollen in their guts. They don't eat pollen. So just to demonstrate, it's the nurses that consume the pollen. And then that protein is transformed into jelly. That jelly is the currency in the hive that is passed from the nurses to the queen, to the larvae, and to the returning foragers that need protein for maintenance. And this passing of jelly and nectar from the crop inform all the bees in the hive as to the colony's nutritional state. So let's go through a year in the hive. First cue to get going is the first tree pollens coming in. Our area is the alder trees. And this starting in January, our colonies start to build up in response to those tree pollens. See here from Manitoba, this is when the trees start to bloom here, and see the brood start to ramp up right here. This is a picture from our area, January, this fresh alder pollen coming in faster than the bees can even consume it. And they start initiating their first brood rearing of the late winter right here. And then these young larvae put off this pheromone, E-beta. I'm sorry, this is fresh pollen. This fresh pollen stimulates the nurses then to feed the queen a bunch of jelly, let her develop her ovaries, and then they will then also feed all the larvae, allow those eggs to hatch and feed the larvae. Then the pheromone from those young larvae, the E-beta ocimene, is a signal to the foragers coming back, whether they should be foraging for nectar or whether they should start focusing on pollen. The larvae are saying, we need a bunch of food, protein, so that's a signal for a number of foragers to start foraging for pollen. Then we get to the spring turnover, where the population of adult bees starts to age and die, and the amount of brood starts to increase. This is from Al Abitabli back in 78 here. And you can see it happening right here. Notice that these winter bees, which have virtually no mortality over the winter, notice that population stays the same. And they get up to, in Manitoba, 200 or more days of age. Once they initiate feeding, acting as nurses and feeding larvae, they shift from this immortal physiology, this diutinous physiology, to nurse bee physiology. and transition to the stages very quickly as the young bees emerge and shift to foraging, and they all going to die off in a month or two right here. So notice how this blue cohorts here just shift down to nothing very quickly. And during this period of time, you may have 2 or more cells of immature bees as larvae and pupae to every adult bee in the hive. Very, very critical time. And the key thing they have to do before they die off is to rear replacement. These are the replacement bees that are going to take the place of those bees. This is often the most, in many areas, the most difficult time of the year for colonies. If you get rain at this time, here in California, we get lots of rain and often snow during this period of time. East Coast, same type of thing. They get these late winter, early spring storms, and that can just kill the colony during this critical spring turnover. If these dying off workers, these old workers, cannot rear replacements, the colony is toast. We see this in the almonds. We take bees in, you take bees right out of cold storage, and they suddenly put them onto major foraging like this. It's the acceleration of their attrition due to the foraging. You can have colonies collapse right there in the almonds that were full of bees. They just didn't have the time to make the proper spring turnover. So here's a comb right here, beautiful comb of brood here of these older bees trying to rear their replacements before they die. And at this time, you can have different pathogens start to cause issues. This is 2-year curve here, but showing how European foul brood and shock brood are typically springtime diseases. Nosema, you're going to see anytime you have a bunch of pollen coming in, you're going to see plenty of nosema. Doesn't necessarily hurt the colony to any great extent. Then during the summer, you typically don't see any kind of pathogen disease other than possibly American foul brood. And then late summer, Varroa and deforming wing virus become big problems. Then during the winter, deforming virus or the other paralytic viruses can be issues, especially with combined with nosema. So if you know your seasonality of disease, you know what to look for at different times of the year. Here's a picture I took of a bee gut, mid-gut tissue. Here's a tracheal tube. Every one of these white dots is a nosema spore. So you can see how nosema just completely takes over the gut tissue. And amazingly, these bees can survive. And I'm always amazed by that. So here's a 3-frame colony, 3 frames of bees in February in our area where we have these frosty nights. And one look at this says, oh my gosh, this poor colony is stretched to the max. They're on the verge of possible collapse because if we're having frosty nights, I know that this many adult worker bees could never have kept that amount of pupae warm over the night. There's not enough bees to cluster over them. So that means that attrition is exceeding recruitment. And these old bees have to keep this brood warm to take their place. If we have one really cold night and much nosema in the hive, in a couple of days, it could look like this. Here's the band of bee bread around. So you know this was solid brood just a couple of days ago in this area. But if you have high nosema prevalence in these bees, they do not cluster, maintain brood very well, fairly high attrition. And if you look at these bees one at a time, their guts, you can have 8 out of 10, 10 out of 10 bees infected with nosema. Here's another typical one. We saw this during the invasion of Nosema ceranae, concurrent with what was called CCD at that time, colony collapse disorder. Very typical nosema collapse symptom. A common misunderstanding is for beekeepers, if they see this dysentery on the front of the hive, they misinterpret it as thinking it's caused by nosema. I've never seen any evidence whatsoever that nosema causes dysentery, and plenty of evidence that says it does not cause dysentery, either Nosema apis or Nosema ceranae. But if the bees are defecating like this inside the hive, that can certainly spread nosema spores, so exacerbate the nosema problem. But dysentery is caused by water balance in the gut, excess water in the rectums of the bees. only way you can diagnose nosema is by microscopy. So here's, this is blowing up from 400x, a pollen grain, typical bee, seta or hair here, and these white oblong objects are nosema spores. This would be nosema ceranae. It's a little bit longer than nosema apis. And the key thing here is nosema ceranae does not normally have much effect on well-nourished colonies these days. But if it has a very high rate, now this is a fairly highly infected bee right here, then if the proportion of bees in the colony is infected is high, then that can slow colony buildup or depress honey production. Okay, to get past this time of year, the spring turnover, if there's lots of pollen coming in, the colonies can grow very, very rapidly. Here's growth curves from Nolan back in 1925. And he shows this linear growth of colonies. Just, man, they just, that 500 extra bees a day, they just take off very, very fast until we reach this equilibrium at about 40 times the queen's egg laying rate. So if a queen's laying 1,000 eggs a day, colony population tops out around 40,000 bees. If she's laying 1,500 eggs a day, you can get colonies up to 60,000 bees or so. And it takes about 60 days of this linear growth before from when the brood nest is established and the colony reaches peak population. You can see this on Lloyd's data right here of this linear growth of the colony right here. Now these colonies can be living hand to mouth. This is from my operation. We had already put the second brood chamber of drawn comb. These are springtime splits that have grown up to be very strong. Of course, we'll smoke these bees off the edges here before we put this box down here. But you can have a colony blooming like this that is living hand to mouth, very small reserves. And there's this huge turnover, very rapid turnover of population. If bees only live 35 days, that means roughly once a month, all these bees need to be replaced. And that takes a lot of food, about 1 to 2 pounds of pollen every single week, just to provide the nutrition necessary for this natural turnover of the worker population. So here's a colony living hand to mouth. They're very minimal amount of pollen right here. And they can do fine. They don't have to have reserve. It's just the problem occurs is if you get poor weather. And then the pollen is depleted rapidly. The nurses will often respond within an hour based on research by Carl Krelsheim, who used sprinklers on hives. They start cutting back on the amount of jelly fed to the larvae within an hour of turning the sprinklers on. And I noticed in Lloyd's, one of the hives that I modeled this from had this dip in brood rearing right here. And when I looked back at weather history from Manitoba for 1976, sure enough, this is when a cold rain storm moved in and the colony immediately responded by reducing brood rearing. And back to that same picture of this colony. These colonies looked just like those wall-to-wall bees. And we had a late April snowstorm that just surprised everybody and many colonies starved. This is survival mode right here. And they've cannibalized most all the brood, and the nurses will recycle that protein back into their bodies, store it to try to survive and or to convert it to jelly. Here's a picture of me treating my bees better than I treat my own offspring. My sons have grown up knowing that springtime in the Sierra Nevada means that you do lots of bee work in foul weather gear. There are many years in the springtime when we dress in foul weather gear 7 days a week for maybe a month if we're having lots of springtime rain to take care of our bees. So we're out there feeding pollen sub to these colonies, brushing the bees off with a pine bough right here. Somebody said, Randy, you guys are out feeding your bees when it's raining and snowing? I said, well, duh, yeah. When it's warm and sunny, they feed themselves. If you're a beekeeper, it's your job to take care of your bees when conditions are poor. And so this is what we do during the springtime. The serious beekeeper is out there working in rain gear every day, taking care of their hives. We also will sometimes, if they're short on stores, on honey stores, we'll simulate a nectar flow by using feeding sugar syrup at that time. Sugar syrup and protein feeding together are the most stimulatory. Hive inspection, quickie. One frame out of the middle of the second brood chamber is often all you need to see. If you pull a frame out like this, you can see at one glance, they have honey reserves at the top. They have their queen right. To be sure, you'd have to see young larvae or eggs, but this is telling me it's queen right. You have very high larval survival. There's very few empty spaces right here. So this pretty much tells you this colony is doing just fine. You don't have to do too much more looking. So healthy brood, high larval survival, spotty brood. Now this could, in the springtime, this would typically be a sign of European foul brood or poor nutrition. If you see this in July or August, that typically just means not enough pollen coming in, and the nurse bees are following the queen and eating the eggs pretty much as fast as she lays them. If you see larvae of different ages like this, that means that you probably have a bacterial or viral disease in the brood, that the larvae are dying and the queen's coming back and laying an egg. So you don't normally see young larvae right next to older larvae like this. So this is a colony that is under some kind of disease stress of the larva. Chalk brood, very typical white chalky looking pupa with this very distinctive oval of tan. This tells you it's chalk brood. It's a fungus disease. No cure for it other than possibly requeening. We see a lot more of this European foul brood, of these third and fourth instar larvae turning yellowish and dying in the cells, and this very symptomatic twisting of the larvae in the cells as they die. We didn't used to ever worry about European foul brood. It just went away with the nectar or pollen flow, but there's some strains now that are not going away. Luckily, all the ones I've seen respond to oxytetracycline, and you spot treat and eliminate the disease from the hive. Generally, if you have a nice colorful palette of mixed pollens coming in, the colony will enjoy good nutrition. And here, look at the step down here of these larvae from the older to the younger, nice even step down in age. That indicates that these larvae are doing fine with lots of jelly on the bottom of the cells. And this is what we do in our operation. We monitor how much jelly is at the bottom of the cells of not so much the older larvae, but these second instar larvae right here. First instar larvae don't get much jelly, but second instar larvae, you want to see the bottom of the cell covered with jelly. So here we go. Here's a abundant jelly larvae swimming in jelly right here. This colony is in great shape, and they can handle almost any kind of external stress. Here's larvae just barely getting enough jelly right here. These will be reared to adult bees, emerge as normal looking workers, but they will not have their full potential. They'll begin foraging at an earlier age than workers that were fed more adequately. So they'll have shorter longevity, so that changes colony population dynamics. Again, larvae swimming in jelly right here, nice healthy colony, and larvae being fed just barely enough jelly to get by. Okay, so now we got that linear growth phase. We're getting up to the swarming impulse. About when the amount of brood in the colony starts to peak out is when the colony will typically swarm. We have lots of sealed brood, especially when you have backfilling, where now you start getting nectar right against the brood, and the colony is restricting the amount of area that the queen can lay eggs in. So if she ceases egg laying for a day, a few days later, there's no more E beta osmine by those young larvae, and the entire colony knows, oh, something's either wrong with the queen, or it's time for us to rear swarm cells and reproduce. Start rearing these queen cells. This is swarm preparation right here. And the drone cells here, there's no sense of them swarming unless there's drones being produced. And you can manipulate the colony by reversing brood chambers here to give the queen heated combs above the brood nest so that she can move up there. And this will This will often reverse the swarm impulse. If it gets to this point where you start seeing all these queen cells with jelly in them on the bottom bars, it's probably too late to do any kind of swarm prevention. You got to make splits. And what we do, we set up assembly lines. And we make about 3,000 nucleus colonies every spring. Otherwise, you get swarming. And you do want to avoid swarming as much as possible for a few reasons. Number one, you lose much of your field force, so you're going to diminish your honey crop. Number two, you now have a competitor out there competing against your hives. And number three, if these are successful in establishing a nest, then they will become a Varroa bomb later in the season and come back to bite you in the butt. So try to avoid allowing your colonies to swarm. Now, if they do swarm, what you can see is this is typical from a colony about to swarm, lots of sealed brood. Notice that the sealed brood is stacked like cordwood, very, very tight. But notice that one adult bee covers 3 cells. So that means that if one frame of sealed brood will emerge to be 3 frames of adult bees. This allows a colony to recover very, very rapidly. And also during swarming, it means that most of the mites remain in the colony, in the parent colony, when a colony swarms. So the colony is really going to be hurting from Varroa after they swarm. Then we get to the main honey flow. And ideally, the colony reaches peak population right at the main honey flow. And because of colony population dynamics, larger colonies make proportionally more honey than do smaller colonies. So we will combine our weaker colonies at the beginning of the main honey flow to allow them to adjust their field force and mid-age bees to more efficiently make honey. So you're better, if you want to have honey, the larger your colonies are, the more efficient they are at making honey. And then the honey flow begins. You can shake the incoming nectar. This was at 10:30 in the morning. That means that nectar all came in just that morning. This is from blackberry. And fresh nectar starts at this at the energy interface. Start getting fresh nectar here, forcing the brood nest down. And the bees shift their priorities. Now their priority is not brood rearing, their priority is honey storage. If they run out of space of drawn comb, not foundation, but drawn comb, then that tells them that they need to start making more comb. And you'll see this whitening of the wax, which is the signal to the beekeeper that the colony is running out of drawn comb and needs more space. And this is the only time you can draw foundation, is when you see white wax. Otherwise, you're just wasting your time. The other thing to understand is the bees are loath to cross a honey band above the brood in order to draw a foundation, especially if there's a queen excluder right here. So if you want to draw a foundation, put it above frames where the brood comes right up to the top bars. And then the bees have all these feedback loops to manage the proportion of mid-age bees, which are your honey processors and comb builders, against your foragers. Roughly, you need to have one one mid-age bee per forager to process that honey. I'm not going to go into the details though. And brood rearing is deprioritized. The reason is once the main honey flow starts, any eggs laid at that part will never, don't have time to become a forager bee and really help. So the colony deprioritizes, and they'll often fill the brood cells with incoming nectar. And you can see this happen in Manitoba when the honey flow started. Bingo, they just cut the queen way down on brood rearing to reduce that. Otherwise, they just have a bunch of extra hungry mouths to feed after the flow is over, which the colony does not want. You can trick the colony then into storing excess honey by artificially enlarging the cavity. Tim Hives here in Idaho or Indiana, he's making 150 pounds of honey, surrounded, and I checked, completely by seed treated corn and soybeans, these huge honey crops. Then in the late summer, population decline, no food coming in, colonies go into survival mode during this pollen dearth. And the attrition exceeds recruitments, the colony population starts going down. the colony does not want to have a bunch of bees here because they're just extra hungry mouths. You see on noise data this drop in colony population, and following this diminution of brood rearing. And your combs often look like that, like this then, with a spotty brood because of the nurses following and just eating the eggs up after the queen lays them. You can, if you wish, if you have reason to do this, don't do it unless there's a reason. So if you're an almond pollinator, you would have a reason to do it. You can rejuvenate your colonies by feeding them protein. These bees will just gobble this up, these nurse bees, and convert this into jelly. This is pollen sub. Now, Varroa. I have a freebie model up there, Randy's Varroa model you can use that will have your colony. You can adjust your colony population, your amount of brood. It'll show you what your mite wash counts will be. You want to avoid this crash. So you can use this to figure out your management strategy for the year. What happens is as the bee population it goes down and the mite population continues to go up late in the season, your infestation rate of the adult bees just explodes right here. It's not the mites that explode, it's your infestation rate that explodes. And a colony can go from 100 mites in the springtime to 10,000 mites by the end of the season. And at that point, the colony starts to collapse. You're going to see, perhaps you'll see some Varroa sensitive hygiene where the bees are removing infested pupae. You may see bald brood. Well, as we breed for Varroa resistance, we're seeing lots more of this bald brood. This is happening worldwide with bees. This seems to be a very good trait for Varroa resistance, where they simply uncap and then recap some of the infested pupae. You may see bees emerging with deformed wings, and you start to see the larvae and pupae dying from virus infection. And the reason is as this is your infestation rate of the bees right here, mites per 100 bees, and this is your prevalence of the viruses, deforming wing viruses, and the paralytic viruses here, and they track right together. So you want to keep your infestation rate down to this very low level where viruses are not much of an issue. Once you get about a third of your worker pupae being infested by Varroa mite, then that's the tip point. The colony will soon collapse. And you're going to start seeing bees unable to emerge, bees with deformed wings up here at the top. And then parasitic mite syndrome, where the viruses start to just take out all the larvae and the pupae. And it looks pretty ugly. And finally, the colony may die, and you see the telltale signs, these fecal deposits from the mites in the remaining brood area. And this is a sure sign that it was Varroa was the problem for the colony. Unfortunately, see Varroa mite here, Varroa mite here. There's a lot of drift of bees from colony to colony. And so highly infested colonies are just mite diffusers to every other colony in the neighborhood. Number one Varroa management strategy by beginning beekeepers is called wishful thinking, and it has never worked. So I would suggest you move on to a better strategy. Typically, in most areas, it takes about 2 to 4 Varroa reductions, however you want to do it. I'm not telling you any method for doing it, but reducing Varroa population by one method or another, typically 2 to 4 times, depending largely upon how many months of brood rearing you have. I've got this on my website showing how we manage our bees in California. I'm not going to go into detail on this now. We are very much involved in a selective breeding program for colonies that exhibit resistance. These are mite counts of 0. So there's your July, August, September, November mite counts. And we say zeros are heroes. We want to see colonies just keep 0 mite wash counts throughout the course of a year. And it looks like we're having some success, but we're not totally mite resistant yet by a long shot. Then in late summer, early fall, the fall pollen flow, the bees will store pollen beneath the brood nest as bee bread. rearing this last generation of winter bees right here. And you can see here where these bees that emerge from this point on are going to age and start forming these long-lived winter bees. And this nice graph from Heather Matala shows this, that the bees this is your survivorship curve of the workers. They die very rapidly all through the summer. But then starting, those that emerge in September and October have this longevity here until they initiate brood rearing here, and then they die again very rapidly after they initiate brood rearing. So what we do in our area, we control the mites first in late summer, and then once we control the mites, because if you're rearing brood, you're also rearing Varroa mites. So get your mite count down first, and then we stimulate our colonies to rear some well-fed winter bees by supplementing them. And this is only because we don't have a fall pollen flow to speak of. Any colony that does not consume their pollen, that's toast to us. We just walk away from them. There's no sense wasting time.
Hey, Randy,
Yeah.
hi. It's a little after 10. I just don't know where you are relative to
No, I'm about
finishing,
about three slides.
Oh, perfect. Perfect, because we do have some questions for you too. Thank you.
Oh, great. And then when pollen stops coming in, you can see the bees just reduce their brood rearing here. No larvae, so these emerging bees will store their fat bodies, protein in them. And then the fall turnover, about half the bees, all the bees, the summer bees fly off, die of natural attrition. Your colony population drops and they go into the winter cluster. Let's just skip through that. And, oh, there's more than I thought. Okay, we're back to springtime again, and the cycle begins again. And okay, that's it. So let's let's go for questions.
Hey, so Randy, I just want to give you my initial reaction. I mean, I just have kind of one word, which was, wow. I haven't heard this talk before. I knew you were giving this talk, but you far exceeded my expectations. And I don't think I'm alone in saying that we will post this talk on our website, and I will watch it many times because you gave us way more information than I expected. I mean, I'm not a brand new beekeeper, but you know, I'm still learning. Like you said, we all learn. But you told me things today that I've never ever heard in many talks. So thank you so much.
I get that feedback from experienced beekeepers everywhere with this talk. You go, man, I've been keeping bees most of my life, and I had no idea.
I didn't even know that bees necessarily cannibalized larvae.
Oh yeah.
So anyway, I'll stop talking. I want to get to our questions. Okay, our first question. Matthew McLean asked, should you see that band of pollen around brood on every brood frame? We see full frames of pollen sometimes right next to the brood, but sometimes not banding the brood.
Yeah, any band, it means that there's a surplus. That's uneaten. That's the leftovers. So on many colonies in the springtime live hand to mouth. There's no band of pollen whatsoever. If there's surplus, that's what's left that's not consumed. The pollen foragers preferentially place the pollen right in the middle of the brood area, and you don't see it because it's eaten immediately by the nurse bees. So the frame to the side of the brood nest or the band is the periphery, the uneaten pollen.
Great. Alyssa Glover asked, how did you diagnose the CCD case as nosema? Sounded like it was due to more attrition, or is it that nosema causes abnormal attrition?
Yes, nosema causes abnormal attrition, and the way to diagnose it, you pick the bees off one at a time, dissect out their gut, you just squeeze their gut out, look at them one at a time under a microscope, and you figure out the proportion of bees out of 10 that are infected with nosema. When you exceed about 6 bees out of 10, the colony starts to go downhill.
Excellent. Okay, our next questioner has 3 questions for you, and only because it's Amy Owen are we going to allow her to have 3 questions. The first question is, pollen is often stored to each side of the brood nest, and I'll often see that they have little or no pollen surrounding the bars or frames of brood.
Yeah.
I'm assuming they carry the pollen from the outside to the inner brood chamber. Is this true?
Right. Erroneous assumption. No movement of pollen ever takes place in a hive.
Okay. If adequate pollen is hugging the brood nest, should a beekeeper still be worried about a lack of pollen rainbows surrounding each brood frame?
Well, first thing, there's no reason to worry about anything. Worry is a waste of time. Understand the biology. What that means is there's a reserve, but it's not stimulatory to the nurses. Pollen coming into the middle of the brood frame is stimulatory to the nurses. The reserve gives them something to fall back on. So if you want the colony to be in growth mode, that's not enough. It's enough for survival mode.
Excellent. Okay, she followed up with, she's in quote, she says, cap and recap infested pupa. So a pupa can be recapped? She says, whoa, I thought they just got pulled out.
No, that's that. Initially, they identified Varroa sensitive hygiene or hygienic behavior, but now they're seeing that we started seeing a lot of this ball brood after a few years after Varroa arrived. And as we breed more and more for resistance, we're seeing much more. But I just took some good pictures of it just this week from one of my resistant colonies. Dr. Stephen Martin communicates with me frequently. He's traveled the world looking at resistant populations of bees, and the one universal trait they have is uncapping, recapping behavior, and not as opposed to freeze kill brood behavior or grooming behavior, which many beekeepers have selected for. But if left to their own devices, it's uncapping, recapping.
Very cool. Okay, Amy's last question. I've read studies that show that a lack of pollen is what stimulates winter bee physiology. You still seem to feed pollen. Does this reduce the number of winter bees produced?
It's not lack of pollen, it's lack of emerging bees that stimulates. And I don't even know if there's such a thing really as winter bee physiology. It's simply nurse bee physiology that is held there because there's no new bees pushing those bees off the combs. It's the emergence of, there's a very good paper published showing how this works. And on my website, look at, towards the end of my series, of Colony Buildup Decline, I've added to that article a few times recently of what's actually happening there. It's the presence of emerging bees that push the nurses off that gets them to shift to older physiology. If there's no emerging bees, then they by default become diutinous bees.
Nice. Okay, well, I'm sure you're going to be listening to our talk from Tina, where she talks about winter bees.
All right.
Okay, good. And I see a question also from my friend, John Triolo, up here in Santa Fe. He's a brand new beekeeper. And this gentleman and his wife, they started with 2 Apame hives. And man, they are just kicking butt, making honey, more honey than I'm making with my hives. So I'm a little jealous. He has a question about mite reduction, but I think we'll save that till your talk this afternoon.
Okay.
So Randy, man, you are a rock star. I am so glad that we invited you back. And I wish we could do some, virtual applause, because I really think that was quite the talk. Really, really good. Thank you so much. I'm glad you're sticking around with us, and great job. Really great. Thank you.
Okay.